Welcome to section 11.1, Discovering How Populations Change. In this section, we'll explore how populations change over time and how scientists develop the ideas that explain those changes. We'll meet early thinkers, see how Darwin and Wallace arrived at natural selection, and connect those ideas to population genetics, the study of how gene or allele frequencies shift from one generation to the next. By the end of this section, you should be able to explain how Darwin's theory differed from earlier fixed species views, describe how today's modern evolutionary theory developed, and show how population genetics uses allele frequencies to study evolution in real populations. Before Darwin, many philosophers and religious traditions framed species as fixed and unchanging. This view was influential for centuries. But as scientists gathered data, including fossils, biogeographical data, and comparative anatomy, alternative explanations started to look plausible and testable, opening the door to evolutionary thinking. While Charles Darwin gets the credit, other scientists proposed ideas to explain species change. Leclerc proposed that species might change after dispersing from an origin point, and Erasmus Darwin suggested that more successful animals would contribute more to future generations. These are early hints of evolution by differential success, even if the mechanism wasn't fully worked out yet. Jean-Baptiste Lamarck proposed that traits acquired during life, like a giraffe stretching its neck, could be inherited by offspring. Although we now know that this mechanism is not correct, Lamarck's big contribution was arguing that natural laws could drive biological change, a crucial step toward a scientific theory of evolution. In the mid 1800s, Charles Darwin and Alfred Russel Wallace, working largely independently, both recognized natural selection. Individuals with traits that help them survive and reproduce leave more offspring, shifting trait frequencies over generations. Their joint presentation in 1858 introduced the idea formally. Darwin's book followed in 1859. On the Galapagos, Darwin noted closely related finches with different beak shapes, each suited to local diets. Some ate hard seeds, some insects, some cactus nectar, and more. From these patterns, he inferred modification from a common ancestor. Island species diverged as they adapted to their specific niches. Natural selection rests on four ideas. First, that traits are inherited. Second, that organisms produce more offspring than can survive. Third, offspring vary in traits. And fourth, that traits that increase survival or reproduction become more common. The long-term studies by Peter and Rosemary Grant on Daphne major showed that after a 1977 drought, surviving finches had deeper beaks that handled tougher seeds. Mean beak size in the population increased measurably, a clear case of natural selection in action. Early 20th century biologists rediscovered Mendelian genetics and unified it with natural selection into the modern synthesis. This explained how small genetic changes, shifts in allele frequencies, could accumulate to produce evolutionary change within and among populations. In population genetics, we often define evolution as a change in allele frequencies over time. The data shown here is a graph of the worldwide distribution of allele frequencies for the A, B, O blood group. Blood type is determined by specific antigens, A and B, present on the surface of red blood cells. Because we inherit one copy of each allele for this gene from our parents, a person can have blood type A, B, AB, or O. A and B represent antigens on the surface of red blood cells, while O represents the absence of either antigen. The positive and negative associated with each blood group refers to Rh factor, which is an additional antigen present, positive, or absent, negative, on red blood cells. We can compare the worldwide ABO blood group distribution with the distribution of blood groups within a specific country to see whether there are any changes. The data shown in the pie chart here are the ABO blood group distribution for Mexico. Differences in allele frequencies like these can reflect population history, migration or gene flow, genetic drift, or selection. Tracking these numbers lets scientists measure evolution and compare populations in a precise, quantitative way. Two processes power genetic diversity. Mutation, or random changes in DNA, is the ultimate source of new alleles. Sexual reproduction reshuffles existing alleles into new combinations every generation. Together, they supply the raw material that selection, drift, and gene flow can act upon. Hardy-Weinberg equilibrium is a null model. If no evolutionary forces act, and if mating is random, then allele and genotype frequencies remain stable across generations. We use the Hardy-Weinberg equilibrium as a baseline expectation. When real populations deviate from it, that's a clue that evolutionary mechanisms like selection, drift, mutation, or gene flow are at work. One common Hardy-Weinberg equilibrium violation is non-random mating. For example, individuals preferring partners with similar phenotypes. Importantly, non-random mating by itself does not change allele frequencies. It mainly shifts genotype frequencies, which often increases homozygosity. That's why it doesn't cause evolution directly, though it can set the stage for selection to act differently. The peppered moth is a classic case study. During the Industrial Revolution, soot darkened tree bark. Dark moths were better camouflaged and survived more often, so their frequency increased. Later pollution controls lightened the trees, and light moths again had the advantage, so the population shifted back. This is natural selection changing allele frequencies as environments change. At first, most people believed species were fixed, a view rooted in ancient philosophy and reinforced by religious traditions. But naturalists like Buffon, Erasmus Darwin, and Lamarck began to challenge this idea, suggesting species could change over time. Charles Darwin and Alfred Wallace took these ideas further. After observing patterns in nature, especially on islands, they proposed natural selection as the driving force behind evolution. Their insight that advantageous traits become more common because they enhance survival and reproduction revolutionized biology. In the early 20th century, Mendel's genetic principles were rediscovered and combined with natural selection in what we now call the modern synthesis. This integration explained how gradual changes in populations can occur over generations. Finally, population genetics gives us the tools to study evolution quantitatively as shifts in allele frequencies within populations. Together, these ideas provide a framework for understanding how populations change and diversify over time.